Okay, hello to all of you. Welcome to my course Biology 1107, and this is the first chapter we are going to discuss in this course. And let me go into the slideshow mode and then start discussing the chapter. So, chapter number 1 is basically a general chapter in which we are going to focus basically on scientific method and evolution. So basically, first of all, let's talk about some of the general things in biology, because this is a biology course. So, and then we will move forward and discuss about what is scientific method, how scientific method is done, and what is basically evolution. So these are the two main components of this chapter. So first of all, what is biology? So biology is a science, it's a branch of science which deals with how life works. So biology can be studied at various different levels. So it can be studied at a molecular level. It can be studied at the level of the molecules which are there inside the cell. And so it's called a molecular level. And then the biology can be also studied at a cellular level, which means how these cells they interact with each other. And then the biology can also be studied at the level at which the cells which forms tissues, and then tissues form organ systems, or they form organs, and the organs form organ systems, and these organ systems form an organism. So how does this organism as a whole interact with the environment? So biology can be studied at various different levels, and these are some of the characteristics of living organisms. So the living organisms are, they are organized, they need energy for living, they reproduce, they respond to stimuli, they are homeostatic, which means they can maintain the internal environment, they grow and develop, and they have the capacity to adapt. So this basically picturizes you the characteristics of living organisms. So you can see the living organisms, they have an order, they are structured, they basically adapt to the environment, they respond to the environment. All living organisms, they reproduce, they grow and develop, they attain energy from the environment, and also they do the homeostasis. So this is basically showing you a boy who is eating an apple. And why he's eating an apple? He's eating an apple because apple is going to give him energy, and energy is required to perform various activities. All living organisms, they reproduce, which means they produce or they reproduce, and they reproduce of their own kind. Like cats give birth to cats, dogs give birth to dogs. And from where does this information comes? What kind of organism is going to be produced? It is there. So why do humans reproduce and form human babies, or dogs produce dogs, cats produce cats? Because this all information is there in the genes, and the genes are made of DNA. All living organisms, they respond to stimuli. So we all respond to stimuli, whether it's a physical stimuli or a mental stimuli. So this is another character of living organisms. Organisms are homeostatic, which means they can maintain their internal temperature, internal environment. Or for example, like as humans, we can maintain our internal temperature. So our temperature stays 98.6. If the temperature goes a little up or a little down, then we are not we have a feeling like we are sick, right? We feel feverish. All living organisms give birth to their own kinds. So you can see this is a plant which is giving birth to its own kind. So organisms, they have a capacity to adapt. So this is one of the very, very important characteristics of the living organisms, that all living organisms, they adapt to the environment. Because if they will not adapt to the environment, they will be thrown out of the environment. And this is also the basis or the principle behind natural selection, which is one of the mechanism of evolution. So according to this principle of natural selection, what does natural selection basically means? It means that the nature selects those organisms which can adapt best to the environment. So in the population, in the population of the organisms, the organisms, they are diverse, they're different. So that diversity is basically, they're different in terms of the genes they have. So the genes are different. So and why in a population of the organisms genes are different? Because of mutations happening or other genetic mechanisms happening. Because of that, they have different genes, and because of that, there is a genetic diversity in the organisms in a population. And the organisms which can adapt best to the environment or adjust best to the environment, they are the ones which survive. And this process is called natural selection. So nature selects those organisms which can adapt or adjust best to the environment. Okay, so these are some of the characteristics of living organisms. Now we're going to talk about different levels of biological organization. So basically, so this is how the life is basically. So life is basically made of atoms. So atoms form molecules, molecules form organelles, organelles form cells, cells form tissues, tissues form organs and organ systems, and organ systems, they form organisms. And the different organisms living in a specific area, that forms a population. And the different populations forms communities. For example, if suppose if you go to some forest, right? So in the forest, you will see a population of some trees, like suppose there are some oak trees or a different kind of trees. So that forms one population. But in that forest, there's not only the trees, but there can be some animals living, like this is deer. So these deers, they're going to form deer population. Then there can be some insects. Then they're going to form insect population. Or there can be some microorganism. There can be some cats. So the different populations living together in a specific region forms a community. And how the communities, how these different populations, they interact with their physical environment, like with water, sunlight, that forms an ecosystem. And wherever there is life, whether it's land, air, or sea, that forms the biosphere. So this is the organization of the, this is the way we can study life at different levels. So this is called different levels of biological organization. Okay, so the basic unit of life is a cell. The basic unit of life is a cell. Now what is cell? That is the simplest self-replicating entity. So cell is the simplest self-replicating entity that can exist as an independent unit of life. Okay. What is a cell? Cell is the simplest self-replicating entity that can exist as an independent unit of life. And there are different kinds of cells. Like you can see, this is the ostrich egg. So this is just one single cell, and it is the largest cell. So these are like skin cells. These are like your neurons, which are nerve cells. They are elongated, and these are your skin cells, which are like flat cells. So the cells are of different kinds. And the most important, there are three important characteristics of a, there are three important characteristics or three important features of a cell: that they can store and transmit information, which means they have information in them, which is in the form of genetic material. They all have a plasma membrane, and they can harness energy from the environment. So they store and transmit information, which is there in the form of genes, and those genes are made of DNA. We'll talk about all these things moving forward. And the DNA, which is the genes, they're made of DNA, and the information which is there in the DNA is converted into mRNA by the process of transcription, and the information in mRNA is converted into proteins by the process of translation. So this is called central dogma. So this is the way how information flows from DNA to RNA to proteins. Plasma membrane is a membrane which surrounds the cell, and it acts as a barrier. So plasma membrane is a membrane which surrounds the cell, and it acts as a barrier And through the plasma membrane, there are a lot of things coming inside the cell and moving outside the cell. So the cell can harness energy, and that energy, so these are the three important characteristics of cells. So they can harness energy. That energy comes either directly from sunlight or it comes from the chemical compounds. So sunlight means, the plant cells, they can make their own food by the process of photosynthesis. So for the plant cells, the energy comes directly from the sun. And for animal cells, because they cannot make their own food, they cannot do photosynthesis, the energy is in the form of the, so they harness the energy from the chemical compounds. Now we need to talk about viruses. Now viruses, so you know that right now the COVID-19, which is a coronavirus, it's a virus. So viruses are a borderline between living and non-living organisms. So they're not completely living organisms. So these viruses, they can store and transmit information. So basically they can, basically it means that viruses, they have their genetic material. So the viruses have genetic material in them. They are covered by a membrane that's called protein. They have around the virus, there's a protein coat. So they have a membrane. But the most important thing which is which makes viruses different from the living organisms is viruses cannot harness energy from the environment. So they cannot extract the energy from on their own. So viruses, when they are outside the living body or living host, they are non-living. Okay. Once they infect a host, so once they infect a host, they hijack the cellular machinery of the host and they start harnessing the energy. So on their own, they cannot harness the energy. So that is why they are non-living when they're outside the living host. But once they get into the host, they become living because they get this ability to harness the energy from the environment. So this is the tree of life. So all the organisms, they can be divided into three domains. They are archaea, bacteria, and eukaryota. So archaea and bacteria, they are prokaryotic organisms. And so we have three domains: archaea and bacteria and eukaryotes. Archaea and bacteria, they are prokaryotic organisms, and eukaryotes are eukaryotic organisms. So what does prokaryotic and eukaryotic mean? So prokaryotic organisms are those organisms which do not have a true nucleus in them, but that does not mean they do not have the genetic material. They do have genetic material in them, but that genetic material is not enclosed in a membrane. So that genetic material is not present in a nucleus. It is there. The genetic material is there inside the cell, and that region is called a nucleoid region. On the other hand, these eukaryotes, they have a well-defined nucleus. So the genetic material in the eukaryotes is present in a special organelle called nucleus. And plants, animals, fungus, they all fall in this category of eukaryotes. So this is called the tree of life. Okay, so this was in general we needed to know about biology. Now we are going to talk about scientific method, and then we will talk about the third part is evolution. So so basically, usually when I teach this class when I teach this chapter in the class, I usually show this picture to the students, and I ask them, what they can see from this picture. So some of the students will say that we can see the bird is singing, the bird is eating. Some students say the bird is helping in pollination. So basically what is happening here, basically my idea of showing this picture to students is I want to make them understand that from the same picture we all make different observations, right? So we all make observations, and observation is very, very important. So basically Observation is the first step in scientific method. Observation is the first step in scientific method. And scientific method is not only done by the scientist in the lab, scientific method is also done by all of us in our daily life. So when we have to solve some problem, then we follow the scientific method. So there are steps of scientific method, and observation is the first step in the scientific method. So first of all, we observe something, then a question comes to our mind. So first of all, we observe something, then a question will come to our mind, and to answer that question which is in our mind will give a tentative answer, or the most probable answer to the question in mind. That is called the hypothesis. And on the basis of hypothesis, we make some predictions, and then we test the hypothesis by doing some experiments or making more observations. And then on the basis of the results of the experiments, on the basis of the results of the experiments, we make the conclusions. So if the so when we perform some experiment and when we get good results, if then we say our hypothesis is supported, right? But if suppose our results do not support our hypothesis, then the hypothesis is rejected. So when the hypothesis is rejected, we need to make a new hypothesis. We have to make a new hypothesis. And as I said, if our hypothesis is supported, then we cannot just jump and say that what my hypothesis was or what I was thinking is correct. So we have to sometimes do many experiments to prove our hypothesis. Okay, so when we do many experiments or we conduct experiments in different directions to prove the same hypothesis, so when our fact or our hypothesis is supported by a lot of evidence, then it becomes a theory. Okay, so when a hypothesis is supported by a lot of evidence, and that evidence comes from the data of the experiments. So we perform many experiments, and when the data from those experiments support our hypothesis, then that hypothesis becomes a theory. Now this is an example of scientific method which we follow in our daily life. So this is a girl, her, you can see her flashlight is not working. So this is her observation that her flashlight is not working. So now a question is coming to her mind that why this flashlight is not working. So that's a question. To answer this question, she can make two hypotheses. So on the basis of hypothesis number one, maybe the batteries are dead. Okay, and so if this is her hypothesis, the batteries are dead, then what she's going to do, she can basically test the hypothesis. And how she can test the hypothesis? So the hypothesis can be tested by replacing the batteries. And what she's doing, she's replacing the batteries, and the battery is still not, the flashlight is still not working. So which means that this hypothesis is rejected. So now she is going to, because this hypothesis is rejected, now she is going to formulate a alternative hypothesis. Now the alternative hypothesis is maybe the bulb is burnt out, and then she has to test this hypothesis. And what she is going to do for that, she is going to replace the bulb, and when she replaces the bulb, and if it started working, so it means this hypothesis was correct. Now these are some of the experiments which were done many years back by some of the very famous scientists, and these experiments basically have used the scientific method. So this is a very famous scientist called Francisco Redi. So Francisco Redi, basically what did he observe? He observed that his main observation was that around a piece of meat, he observed many maggots. Okay, so maggots are basically the young ones of these flies. So he observed that around the piece of meat there was there was these maggots were coming, and these flies were coming, and they were laying eggs, and these maggots were forming. Now the question came to his mind was that these maggots, from where these maggots are coming? Are these maggots coming from the environment, or these maggots are coming spontaneously from the piece of meat? Okay, at that time, the scientists did not knew that life comes from life. Scientists at that time did not knew that life comes from life. So basically, so this was a question. So from where does these maggots are appearing? Are they coming from the piece of meat itself, or they're coming from the environment? So what this scientist did, he took 3 jars and he kept one jar open. The second jar was covered by gauze, and the third is totally sealed jar. Okay, so in this jar, this is open, so the flies can come in contact with the piece of meat. So the flies as well as air can come in contact with the piece of meat. The jar which is covered by gauze, through the gauze, the air can go inside the jar. The air can go inside the jar, but these flies cannot. And third one is sealed jar, so neither air nor flies can go inside the jar. So this is the this is the way the experiment was set up. So he made the observation, then then a question came to his mind, then he formed the hypothesis. So on the basis of the hypothesis that the hypothesis was life comes from life, and to test that hypothesis, he performed the experiment in this way. He took 3 jars, and he took one open jar, one sealed jar, another is gauze covered jar. Okay. So what was his observation? The observation was that the jar in which the flies were coming in contact with the piece of meat, there was a formation of maggots. So this jar, which is covered by gauze, the air is able to come in contact with the piece of meat, but not the flies. So there is no maggots forming here. This is a sealed jar. In this jar, the piece of meat is neither coming in contact with flies, nor it is coming in contact with the air. So no maggots are forming here. So this experiment proved that his hypothesis was correct. Life comes from life. So which means that this piece of meat has to be in contact with these flies for the formation of maggots. And this is another experiment, very typical experiment using, this is another problem solved by another very famous scientist called Louis Pasteur. He also used scientific method to solve this problem. So what his observation was that whenever he was taking a sterile broth, he found that the sterile broth, after some time when he was keeping the sterile broth in air, it was getting bad, it was getting contaminated, it was getting cloudy. So again the question was, so when it is becoming contaminated or it's becoming cloudy, so which means that it is getting contaminated with the microorganisms. So from where does these microorganisms are coming? Are they, is it some magic happening? Is it some spontaneous generation of the microorganisms in the broth? Or these microorganisms, they are coming from the air. So if the microorganisms, they're coming from the air, which means that there is no spontaneous generation of life, but life actually comes from life. So this was the observation, that the broth was getting contaminated. Question was, from where this growth, from where does this bacterial growth is coming, from where does this contamination is coming? Either it is coming from the broth itself, or it is coming from the environment. So this was the question. The hypothesis he made was life comes from life. Life comes from life. So for that, what he did, he took a flask which is straight necked, and second flask he took was S-shaped. And in this flask, which is S-shaped, here is a kind of a trap, and there's like a cotton trap here, and the organisms which are there in the air, they get trapped here. Okay, so so basically he took two flasks. One is straight necked, another is S-shaped. The S-shaped has a trap here, and he made the broth sterile. And after some time, what he observed was that the broth in the S-shaped flask, that is getting contaminated, but not in this one. So what is happening is, in this flask there are all organisms here. They come in contact with the broth here and make the broth contaminated. And this is the S-shaped flask, because in this there is a trap here. The microorganisms, they get trapped here. They just cannot go and reach the broth here. So that is why this broth stays sterile. There is no contamination happening here. So this experiment again proved that life comes from life. So this is the experimental setup, and then after you perform the experiment, the results are analyzed and conclusions are made. Okay, so now we are going to talk in more detail about the steps of scientific method. First of all, observation. So as I said, we all observe. So the observation can be done by making use of our senses. Like for making observation, we can use our eyes, we can use ears, or we can also use some equipment. Like in the lab, we make use of some equipment like microscope or some calipers. We use different kinds of equipment to make the observations. Hypothesis. So hypothesis is basically the most probable answer to the question in mind, or it is the most tentative explanation to the question in mind. That is what hypothesis is. And hypothesis, how hypothesis is formed? Hypothesis is formed by using inductive reasoning. Now, what is inductive reasoning? So inductive reasoning is basically making use of the creative thinking to combine the isolated facts into a cohesive fact. So basically, inductive reasoning is used to make a hypothesis, and it makes use of creative thinking to combine the isolated facts and to make one fact out of that. And the hypothesis should be testable, and it should be presented as a falsifiable statement, which means you can prove it. Should not be, you cannot make such hypothesis which you cannot prove wrong. And hypothesis, you cannot make a hypothesis which you cannot test. Suppose if I say that, oh, my flashlight is not working because maybe there are some aliens coming from some other world, or there are some supernatural powers, they are just messing up with my flashlight. We cannot make this kind of hypothesis because we cannot test this hypothesis, right? So we cannot test this hypothesis. So this is not a good hypothesis. So when you make a hypothesis is also influenced by your personal experiences. So to test the hypothesis, we perform experiments, and for testing the hypothesis, the scientists make use of deductive reasoning, and the deductive reasoning makes use of if and then logic. So according to the if and then logic, so basically it means that if suppose, let me give you an example. If the organisms, they are made of cells, then we will be able to see cells under the microscope. Okay, so this is the way we test the hypothesis. So to determine how to test a hypothesis, scientists use the deductive reasoning, and deductive reasoning works on if and then logic. And to test the hypothesis, scientists, they conduct experiments, and for conducting experiments, first of all, we have to design our experiment. We have to first of all sit down and plan our experiment. That's called experimental design. And in every experiment, we must keep a control. Okay, and control has to be included in every experiment, because control is something with which you will be able to do some comparison. So a control is something which is not getting that specific treatment which we are trying to test. So If we do not have the control in the experiment, then we cannot say for sure the effect is of because of what we are trying to test. So for example, suppose you are testing a fertilizer, that this fertilizer helps in the growth of plants. So if you just take a plant and put the fertilizer and your plant grow, then you have nothing to compare with, right? You do not know the growth. You do not know the growth of the plant is because of a fertilizer, the new fertilizer, or what. So for this kind of experiment, so actually in every experiment, in every scientific experiment, we have to keep a control. So control is something which is not getting that specific treatment, and control is used for comparison. So in this experiment, we will take a pot, we will grow a plant, but we are not going to add the fertilizer to this. We are not going to add the fertilizer to this. So then we can compare in the second pot if that effect is because of the fertilizer. Because we are going to compare our test pot. We are going to compare our test pot with the control pot. So the test pot is getting the fertilizer and the control pot is not getting the fertilizer. And scientists also make use of models, like computer modeling is used to study climate changes. Mice models are used for doing cancer research. And when we perform the experiment, we produce the data, and the data can be qualitative data or it can be quantitative data, and the data, we basically represent the data in the form of graphs and the tables, and we have to also do some statistical analysis on the data to see if the data is significant or not. Conclusion. So after the experiment is done, we make a conclusion. We conclude whether our hypothesis is supported or it is rejected. And as I said, when you make a hypothesis and you're trying to see if your hypothesis is correct or not, and when you perform many experiments in the same direction to prove your hypothesis, and if your hypothesis is supported by lot of experimental data, if your hypothesis is supported by lot of experimental data, then that fact becomes a theory. There are many theories in science, but in biology, the very important, very famous theory is cell theory, and also there's another theory, theory of evolution, which we're going to talk about very shortly. And the experiments which involve controls, they're called controlled studies. And in the controlled studies, there are two groups. There is a control group and there is an experimental group. So the control group is that group that does not receive any treatment. So control group is that group which does not receive any treatment, and experimental group is that group which receives the treatment. And the two more definitions which you need to know. One is independent variable, another is dependent variable. So independent variable is that variable which a researcher controls. So it is something which we are trying to study, the effect of something which we are trying to study. For example, I gave you example of the fertilizer. So if you are trying to see the effect of fertilizer on the plant growth, then the amount of fertilizer you're using is an independent variable. And in response to the amount of fertilizer we are using, so in response to the amount of fertilizer we are using, how it affects the growth of the plants. The growth of plants is the dependent variable. So there are two variables in the experiments. One is the independent variable, and second is the dependent variable. Independent variable is that variable, it's that factor which we are trying to study in the which we are trying to test in the experiment. And dependent variable is that variable which changes in response to the independent variable. Okay, the last topic we are going to talk about is evolution. So in biology 1107, we do not talk much about evolution because that's a part of the course biology 1108. But I will just give you some important concepts about evolution. So basically, according to the theory of evolution, the organisms are modified descendants of common ancestors. So according to the theory of evolution, we have all evolved from common ancestors. So according to the theory of evolution, we have all evolved from common ancestors. And how we became different? We became different because of some heritable changes which took place in us. So organisms are modified descendants of common ancestors. Evolution explains pattern of unity and diversity in living organisms. Similar traits among organisms are explained by descent from common ancestors. Differences among organisms explained by accumulation of heritable changes. So we have a lot of things common in us because we have originated from same ancestors. But we are also different. So why we are different? Because some of the changes took place in our DNA, and those changes are called heritable changes. So we are different. So the differences among organisms are because of the accumulation of heritable changes. So this basically talks about that there are like more than 10 million or now maybe it's 100 million species, and basically those species, they have to be organized. So there's a branch of biology called taxonomy. Is a branch of biology that names and classifies species into groups of increasing breadth. So there are different categories of the we can divide these species into different groups. The broadest is the domains, which we just discussed in the tree of life, that there are 3 domains followed by kingdoms. So this is the way the organisms can be divided into different groups. The broadest is domain, then follows by kingdom, phylum, class, order, family, genus, and species. So as we are moving this side, we are becoming more and more specific, and on this side it's the more broad group. We have already talked about the 3 domains of life. So there are bacteria, archaea. These 2 domains, bacteria and archaea, they are prokaryotic organisms, which means they do not have the nucleus in them. So these are the bacteria, these are the archaea. So bacteria and archaea, they're very similar, but they are structurally they're similar, but biochemically they're different. Because these archaea, they can resist very harsh conditions. They can stay in very acidic conditions. They can survive in high temperatures, even in very high salt concentrations. And these are eukaryotes, which includes 4 different kingdoms, which includes plantae, animalia, fungus, and the protist. Okay, so the biological world is very diverse, but there is unity in the diversity of life. So there is a unity in the diversity of life, and that unity in the diversity of life is at the DNA level. So which basically means that all living organisms, they are diverse. But they have something common in them. All living organisms, they have DNA in them. And there is also unity, which is evident in many features of the cell structure. So you can see that this is the cilia. So the paramecium, which is a unicellular eukaryotic organism. So paramecium, which is a unicellular eukaryotic organism, it is all covered by cilia. And the same kind of cilia, it is also there on our epithelial cells, those epithelial cells which make the respiratory tract. So the cilia which surrounds the paramecium is the same kind of cilia is there which surrounds the epithelial cells. So the cilia which is around the paramecium cells, as well as which is around the epithelial cells in our respiratory tract, they're same. Unity in diversity is also evident at the floral pattern in some of the flowers. So fossils are the evidence that life has evolved. Basically, the theory of evolution is given to us by a very famous scientist called Charles Darwin, and he wrote a very important book, and the book's name is On the Origin of Species by Means of Natural Selection. And Darwin made 2 points in this book. Species showed evidence of descent with modification from common ancestors, and natural selection is the mechanism behind the descent with modification. So this is the picture of Charles Darwin, and so basically these are some of the observations made by Charles Darwin. So basically he observed that individuals in a population they vary in their traits, many of which are heritable. More offsprings are produced than survive. So when there are more organisms, when there are more offsprings in the population, and the resources are less, then there is a competition. And species, they generally suit their environment. So basically, in the population, when there are different organisms, they vary in their traits, and those traits are heritable. And if there are more offsprings produced, less resources, there's going to be competition. And then what is going to happen? Nature is going to select those organisms which can adjust best to the environment. Darwin inferred that individuals that are best suited to the environment are more likely to survive and reproduce, and over time more individuals in a population will have the advantageous traits. So basically the organisms which are going to be selected by the nature on the basis of the characteristics, they are going to be the ones which are going to survive, and once they survive, they reproduce. So that means that those organisms, they are going to propagate in the population. So this is what is the concept of natural selection. So this is an example that you can see. These are the insects, and these insects are sitting on a soil, and that soil is has a dark color. The soil has the dark color, and these are the insects. And these insects, they have the skin is of different colors. Some are dark, some are light. So this bird is going to eat the insects. So this bird is going to eat the insects which are light in color, because they are visible to the bird. And the insects which have a dark covering, they basically merge with the background, so they cannot be seen by the bird. So what's going to happen in that case? So in that case, the insects which are light in color, they will be visible to the bird, and they will be eaten by the bird. So which means that the insects which have which have light covering, they will be eliminated from the population, and the insects with dark covering, they are going to persist. They are the ones which are going to persist, because they cannot be seen by the bird. And the wings of the bat, this is another example of natural selection, because bat is the only animal which has webbing in between the bones. Okay, so basically, maybe these bats were in some environment where there was a need for them to fly. So that is why in the population of the bats, those bats which had this advantageous character of having the webbing between these bones, this character was chosen by the nature. So that's a natural selection. So in a diverse population of bats, the bats that had this character of having the webbing between these bones was selected by the nature, because there was a necessity for these bats to fly. So unity in diversity rises from design with modification. This we can say from this example, that if we compare the forelimb of bat, which I just showed you here, this is a forelimb of bat, and if we compare it with the forelimb of human, horse, or the whale flipper, um, they all share common skeleton architecture. They all share common skeleton architecture. Um, so, which basically means that they have a common skeleton. They have a common skeleton architecture, but the only bats, they have the webbing in between. And also, Darwin proposed that in a population, that the different individuals, they attain different characteristics because they are in the different environments, and as a result of that, that results in the formation of different species also. So you must be knowing, Darwin did a lot of work in the Galapagos, Islands, near in the South America. So he worked on the finch species. Finches are basically birds, and he studied these finches in the Galapagos Islands. And what he found, that in the different islands, there were different conditions. There was different kind of food available, and because of that, the finches, they had different beak structures. So these finches, they had different beak structures at different islands, because in the different islands, the availability of the foods was different. Like in some islands, the food was hard. In some of the islands, the food was soft. So on the basis of that, these finches, they developed different beak structure, and that also result in the formation of different species of the finches. So that's all what we need to know from this chapter. And basically, if you see D2L, I have also posted study guide for this chapter, as well as I have also posted one video on evolution, and I have also posted in-class quiz. So basically, that quizzes I usually conduct in the class. So those are practice questions for you. And I will also be posting the Mastering Biology for the first chapter. So get yourself enrolled in Mastering Biology, and Mastering Biology questions are your homework, and they will be very useful to you for your upcoming online quizzes and your test and all that. So all from chapter number 1. If you have any question, please let me know.